Hello everyone, my name is Joan Casals and I am a lecturer at the Polytechnic University of Catalonia and a researcher at the Miquel Agustí Foundation. In the following two short presentations, I will discuss about the phenotyping of the fruit shape traits in tomato. When it comes to plant phenotyping, seeing a wide range of variations for each trait is crucial. It helps us understand the full spectrum of possibilities. In this image, you can see different fruit shapes commonly found in tomato land races from Catalonia. Notice that square, highly ribbed, and hollow fruits are enriched in this collection. Personally, my tomato collection initially had limited diversity. However, working with the Traditon collection, which has 1,800 accessions, expanded my understanding. I discovered new phenotypic variations that gave me more insights into tomato phenotyping. In short, exploring the full variation for each trait in tomato is necessary for effective phenotyping. What is clear is that fruit shape, together with fruit size, have been among the most selected traits during the tomato history, both in the primary and secondary centers of diversity. Variation for fruit shape was, for sure, a trait desired for many farmers around the world, bringing some beauty to the tables. We can see in this image how flat and highly ribbed fruits were very present in Spanish tables on the 18th century, but also how Pablo Picasso painted, surprisingly, brown fruits in a cubist landscape. Several works have shown, since more than 20 years, that the diversity of tomato fruit shapes is primarily regulated by four major genes. Of them, SUN and OBEID control fruit elongation, whereas physated and local number are two genes that control the number of locules or cavities inside the fruit. The latter have also pleiotropic effects on fruit size. Most of the diversity in fruit shape that we can see is the result of the combination of the different alleles in these genes, as it is shown in this figure from the work of Gustavo Rodriguez and collaborators. More recently, different works have unveiled other genes that have important effects on the tomato fruit shape. This is the case of the extreme elongated phenotype caused by the overexpression of SUN, also the recently reported glove gene that is responsible for the spherical phenotype, and finally, Jen Wen-Song and collaborators described the pointed tip gene, which regulates the protuberance of the tomato fruit tip. In this first part of the presentation, we will take a look on the following traits: fruit length, fruit width, the ratio between the length and the width, and the fruit shape in longitudinal section. The length and the diameter and the ratio are crucial traits that determine the fruit external shape of the tomato. In fact, we can consider that all the fruit shape categories are the result of different length to diameter ratios. We can see this interpretation in the figure proposed by CPVO, where the fruit shapes are classified according to if they are wider than tall, which corresponds to the categories very compressed and moderately compressed, if they show similar width and height, which corresponds to the category medium, or if the fruits are taller than wide, which corresponds to categories moderately elongated or very elongated. Despite this qualitative classification is very informative, we can obtain a more precise information if we phenotype quantitatively the width and height of the fruit. As you can see in this image, different estimates of the width and length can be obtained. They can vary, especially in non-round fruits with highly depressed shoulders. With regard to the fruit length, we can either obtain the maximum length, this is the distance between the stems of the fruit, or the length between the stem and blossom end. With regard to the fruit width, there is always the difficulty to decide at which height measure it. We can have, thus, the width at mid-height or the width at the maximum diameter. For most of the cases, there will be a strong correlation between both measures. In our experiment, we will measure the maximum height and width, as it is shown in this figure. Despite these measures are easy to obtain, our main interest is generally to classify our varieties in different fruit shape categories. In this sense, fruit shape in longitudinal section is the most distinguishing trait, and is often the first characteristic that farmers and breeders use to identify different varieties. CPVO proposes a fruit shape classification with 11 levels. These are flattened, oblate, circular, oblong, cylindric, elliptic, cordate, ovate, obovate, piriform, and obcordate. These categories are representative of the tomato diversity. However, it is very difficult to assign one genotype to a single fruit shape class. The main problems that arise when assessing the fruit shape trait are that the fruit shape shows a continuous variation and there is some overlapping between categories. Also, it exists some degree of intra-plant variation for fruit shape. For instance, on big and ribbed fruits, fruit from the first truss can show abnormal shapes. Finally, some categories are difficult to distinguish by the human eye. An easier system to use is the one proposed by Sofia Visa and collaborators in their work published in 2014 in Ophitica. Based on morphometric data and Bayesian classification, these authors proposed a classification based on 9 classes. These are distinguishable by the human eye, as you can see in the image. The main classes are the following. Round fruits. This is fruits that have the same width and length. Rectangular fruits. This is narrower fruits that show a more or less constant width along the longitudinal section. The width-length ratio is close to 1. Ellipsoid fruits. This is fruits that show a much higher length than width, and which show a slight narrowing of the width at the top and bottom. We could observe the profile of the fruit as an ellipse. Flat. This is an easily recognizable shape, which includes fruits that have a much larger diameter than the height of the fruit. Obovoid. This represents an egg-shaped fruit with a narrower area at the stem end. Ox-haired. The fruits categorized as ox-haired tend to be large, showing a significant reduction in width at the blossom end, and tapered with prominent shoulders. Long rectangular. This shape is similar to the rectangular shape, with a more or less constant width along the longitudinal section. Unlike the rectangular shape, the length of the fruit is much greater than the width. Haired. This shape has similitudes with ox-haired, but is present in smaller fruits that are larger toward the proximal end than the distal end, show less prominent shoulders than ox-haired, and had a distinct tip at the distal end. And finally, long, a class that englobes fruits that show a rectangular shape, without shoulder depression and that display a pointed tip. An important issue is that the most representative fruits for fruit shape can be found from the second to the fourth trusses, which are normally used in tomato phenotyping. Coming to the end, a good example of the importance of fruit shape in the genetic diversity of the tomato was recently described by Clara Pons and collaborators. In their study of a panel of 1,800 traditional European tomato varieties, they show how shape and culinary use are the main discriminating factors between land races. The study also allowed the varieties to be classified into 8 clusters, which, as you can see on the right of the slide, represent the different shapes of the fruit that we have just seen. Last but not least, the tomato analyzer. You may know it, but just in case, the research group of Esther van der Nat provides a free tool to analyze quantitatively fruit shape traits in tomato. This tool just requires that we scan the tomato using a home scanner and calculates several traits. The parameters related to the length and diameter are calculated with very good precision. For the shape of the fruit, the program calculates the morphometric points of the perimeter, which can be used to infer the shape. An excellent tool for jobs that require precision. So that's all on my part. I would like to thank the Maritza Vegetable Crops Research Institute for the organization, the HARNESSTOM project, and the Roxy Cost Action for supporting this training course. Thank you for your attention and enjoy the presentations.